So today we discuss glycolysis, which is one of the very important processes where we talk about a process where lysis is involved. But before going to that point, there are some concepts that we need to revisit. So when we talk about respiration, this is a chapter, respiration in higher plants, and glycolysis is an important and a very fundamental metabolic process which occurs in aerobic and anaerobic conditions. Okay, the word glyco and then the word lysis. So this is sugar, a sugar substrate, and breakdown. So breaking down of sugar is glycolysis, and this is definitely giving energy. So at the end of glycolysis, the cell derives energy. Now, coming back to respiration. So, respiration is a physiochemical process. And why is it called as physiochemical process? Because again, there are two parts in respiration that occur. Very important is, we all know, it's exchange of gases in respiration, and then this is oxidation of food. Okay, so that's what composes respiration. Again, it's a metabolic process. Now when we talk about metabolic processes or metabolism, basically, I'll just write metabolism. It is composed of anabolism and catabolism. So these two important processes or phenomenon that happen in our body, anabolism and catabolism, they both combine, and that's what is metabolism. Anabolism is making, formation. Catabolism is breaking. Okay. Now respiration, again we come back to respiration. There is aerobic respiration and then there is anaerobic respiration. We're very well aware of these terms. So again, presence of very fundamental O2. So the respiration that happens in presence of oxygen is aerobic respiration, and then which is happening in oxygen deficit of O2, or say absence of, is anaerobic respiration. So here complete oxidation happens. Here incomplete oxidation happens. So who's incomplete oxidation? Of course the substrate, of course the glucose is completely broken down, and in the aerobic, and in the anaerobic, sorry for this, and in the anaerobic there is incomplete oxidation. So again, like aerobic respiration is going to yield CO2 plus H2O, whereas anaerobic respiration can again be of two types, alcoholic and then the fermentation, which does not yield alcohol. So in alcoholic you get ethanol, and in the fermentation type of anaerobic respiration, lactic acid. So since aerobic respiration is complete oxidation, the yield of energy is high energy yields. Here the energy yield is lesser as compared to the aerobic respiration. So these are some very basic concepts before we start glycolysis. We also know that the breakdown of glucose. When we talk about photosynthesis, we know that it is making of glucose, whereas now in respiration we are talking about breaking of glucose. Okay, now what is glycolysis? It is a process in which the glucose is broken down and energy is given out. So you can say, is it an exothermic or is it an endothermic? It is both actually. It's a multi-step process. A part of glycolysis is endergonic, that is what we call, is basically requires energy. A part of glycolysis, the rest of the glycolysis, is energy yielding, generating. And what is the end product? Two pyruvate molecules. We get ATP, we get NADH, and we get water. The process, where does it occur? It occurs in the cytoplasm of a cell, and it does not require oxygen. So there's no compulsion that oxygen should be present. It can happen in both the situations. Again, it happens in both aerobic and anaerobic organisms. Now, there are some bullet points here. It is a primary step of cellular respiration. It is universal. It occurs in all the organisms. Followed by glycolysis, the pyruvate enters into the Krebs cycle. That is again a purely aerobic respiration, very different from glycolysis. In absence of oxygen, the cell will make ATP, but the ATP amount is very small, because in absence of oxygen, either the cell will switch on to alcoholic respiration or it will switch on to fermentation if it is limited oxygen. This pathway was discovered by three German biochemists, Gustav Emden, Otto Meyerhoff, and Parnas, in the early 19th century. It is also called as the EMP pathway. Moving on to the main multi-step glycolysis process. Now we start with glucose. So glucose is our starting material here, the substrate that we can call as, and this glucose now undergoes a lot of series of reactions, you can see, and this is a multi-step process. How is it? Is it very fast? Is it very slow? So basically it's a slow process. It's a multi-step process. Okay, why is it a multi-step process? In biochemical pathways, it is very important to understand one thing, is that suppose you start with A. Okay, A is your starting molecule. The next step, it becomes B. The very next step, it becomes C. In the consecutive step, it becomes D. So now A was your substrate. B is the next form of that substrate. Okay, it has been, the reaction has occurred and the product is formed. Now this B is the starting material for C. So for C, B is the substrate. Again, for D, C is the substrate. So it's a chain of reactions, and it is extremely important that each step happens properly, because if there is any problem in one of the steps, the next step will not happen normally. And if the biochemical pathway happens properly, occurs properly, goes from one step to the other step in an optimal condition, making optimal concentration of molecules, it is only then it is beneficial for the cell as well. Okay, so now glucose, 6 carbon, it's a 6 carbon molecule, and in the very first step, you see there is a consumption of ATP and ADP is formed, and glucose 6 phosphate. So addition of phosphate group to the glucose at the sixth position, and that's why it becomes glucose 6 phosphate. Now this is mediated by hexokinase. It's this whole pathway, or whatever pathways that you come across in biochemistry, are all enzyme mediated. Enzymes are biocatalysts. Enzymes are important because they enhance the rate of reaction, enhance the substrate conversion into a particular product, and they are reusable. They can finish one reaction and they can again participate into the uh, other reaction as well. So they, we have all studied these the enzymes and all that we have studied in the previous chapter also. So here it is hexokinase. Hexo is six. Kinase, what do you mean by kinase? It is an enzyme that catalyzes the transfer of a phosphate group from ATP to a specified molecule. So you can see here the ATP is, the phosphate is taken out of the ATP, so it becomes ADP, that is adenosine diphosphate, and that phosphate that was removed is linked or is attached now to the sixth position or sixth carbon in the glucose, and it becomes glucose 6 phosphate. The second step is conversion of glucose 6 phosphate to fructose 6 phosphate. Okay, now here again there is one more enzyme that is involved, which is, sorry, not here, it's here, phosphoglucoisomerase. One very important thing to remember is that glucose and fructose are isomers of each other. I hope you all know the structure. If not, we can quickly revise the structure here. So 6 carbon, so this is the glucose, okay, so this is the glucose, and these are isomers. So this is an isomer of fructose, or fructose is an isomer of glucose. So they can convert into each other. If you look at the molecular formula, the molecular formula of both of them is C6H12O6. So what is different is the structure, and you can very well see it here. CH2OH, and then you have H. Okay, so this is fructose, and this also is C6H12O6. C6H12O6, they are isomers, and when they convert, or they, yeah, when they convert into one form, from one form to the other form, again that is enzyme mediated, and here the enzyme involved is isomerase. Okay, so everything, you see that every step is possible only because of enzymes. So here also in the second step also you have an enzyme which is called as phosphoglucoisomerase, also because there is a phosphate group involved. Okay, then glucose is transferring, or glucose is converting into fructose, so hence phosphogluco and then isomerase. So you finish the second step. Now you move on to the third step. Here the fructose 6 phosphate, again not photophosphorylation, phosphorylation of ATP to ADP happens. Okay, phosphate group is added to the first position of first carbon also of fructose. So now the fructose 6 phosphate becomes fructose 1,6 bisphosphate. Now because there are two phosphates here, okay, so this is also mediated by one of the very important enzymes which is known as phosphofructokinase. Phosphofructokinase, it is an allosteric enzyme and one of the very important enzymes in glycolysis, because it can actually regulate the glycolysis, because it can take an account of what is the concentration of ATP right now in the cell, whether the cell is in deficit, whether the cell has optimum and required amount of ATP already there, should the cell undergo, should the cell perform glycolysis. All of these decisions, probably very important decisions, are taken by the PFK. And so here this is the important enzyme. Okay, So for phosphofructokinase, after that, now we have seen three steps here. So this was the step number one, step number two, step number three, and this was the fourth. In the first three steps, ATP is consumed. So step number one, two, three, ATP consuming. Okay, so glycolysis can be divided into two parts. The first part is from reaction 1, 2, 3, and the rest is the remaining of the glycolysis. So in the first three steps, ATP is consumed, and in the rest of the steps, we will study ATP is generated. So these are the two phases of glycolysis. It's a slow multi-step process, and you see that it is an enzyme mediated pathway. All the even in the Krebs cycle, we are going to see there are many important enzymes which are involved there. So now after the formation of fructose 1,6 bisphosphate, which is again a 6 carbon compound or molecule. So we started with a 6 carbon molecule, and at step 4 we are still with a 6 carbon molecule. Now this 6 carbon molecule, it splits into two molecules of 3 carbon. Okay, the first is triose phosphate, which is also glyceraldehyde 3 phosphate, and the second one is triose phosphate. Again, of course, it's the same because they are 3 carbon, but this compound is dihydroxy acetone phosphate. So sometimes we can write it as dihab here just to remember, and you can also call this as PGAL, phosphoglyceraldehyde. Now, this reaction is mediated by another enzyme which is called as aldolase. So you can see that there are two products that are formed. Now that 6 carbon is split into 3 carbon, 3 carbon, and you can see this reversible sign here. It means again they are isomers, and here there is isomerase which is in picture. Okay, now what happens is um, dihap also converts into 3-PGAL. Okay, so it means you had PGAL here, you had dihap here, and they are interconvertible, but dihap converts into PGAL, which means you are now having two molecules of phosphoglyceraldehyde. So that's why this comes here 2 into triose bisphosphate. Yeah. So after the interconversion, you have two molecules of PGAL. Now, here interestingly, the NAD plus is converted into NADH plus NADH plus. Okay. So with this involvement of NAD plus, we see that we get two molecules of again triose bisphosphate here. It was triose phosphate initially, now it is triose bisphosphate, which tells you that one more phosphate group has been added. Initially the phosphate group was at the third position, now at the first carbon also the phosphate group is added. So you have a molecule which is 1,3-bisphosphoglyceric acid. Here the aldehyde is converted into acid. Okay, and this is only possible because of oxidation. So NAD plus oxidizes and we get NADH plus H plus. Okay, two molecules of glyceraldehyde 3 phosphate got converted into two molecules of 1,3-bisphosphoglyceric acid. This is a conversion of aldehyde to acid. It is only possible because of oxidation. Okay, now this 1,3-bisphosphoglyceric acid uses it converts into two molecules of 3-phosphoglyceric acid or phosphoglycerate, whatever you want to call it as. It's a 3 carbon compound. Again, it gets converted into a 3 carbon compound. We started with 2, so we'll get 2 here. ADP is converted into ATP here. Okay, so it means that 1,3-bis phosphoglyceric acid may say one phosphate is released. That phosphate is then taken up by the ADP and ATP is formed. Now we have 3 phosphoglyceric acid here. Later on, this 3 phosphoglyceric acid is converted into phosphoglycerate. Okay, there are 2 molecules of 2-phosphoglycerate. This phosphoglycerate further converts into phosphoenol pyruvate, and phosphoenol pyruvate converts into pyruvic acid. Here also you'll see that ATP is converted into ATP because one phosphate group has been added there. Okay, so at the end of the glycolysis pathway, you get 2 molecules of pyruvic acid. So the net gain in glycolysis in terms of ATP is 2 ATPs. Okay, so glycolysis may you get an yield of 2 ATP molecules, and you can see that you started with glucose and you are getting an end product of pyruvic acid. Intermittently, glucose got converted into glucose 6 phosphate. Glucose 6 phosphate got converted into fructose 6 phosphate because glucose and fructose are isomers. So my question is that if you change the sugar, so if you change your starting material, which was glucose, if you change it to fructose, will the EMP pathway proceed? Yes, it will. Then it will skip the first step. Okay. So fructose also, glucose and fructose both can drive the glycolysis pathway. Okay. And there are few reversible reactions, there are few irreversible reactions in this cycle in the glycolysis pathway. Sorry. So there is something which is called as a rate limiting step. When the irreversible reactions are involved, there is no coming back. So a product or an intermediate is converted into the next intermediate. The next intermediate cannot flip and become the previous one. That is the irreversible reaction. But there are reversible reactions where suppose A and B are interchangeable. B can become A. Okay. At that point of time, the glycolysis pathway will not proceed, or it will become slower, or it will take a pause. So such decisions are dependent on, say, the enzyme which is monitoring. So phosphofructokinase is very, very important here. And in the third step of EMP pathway is also the rate limiting step. Okay, so aerobic respiration, anaerobic respiration, alcoholic and lactic acid fermentation, they are from anaerobic respiration. Aerobic respiration, we're going to see the Krebs cycle also. Glycolysis has no, it is not mandatory for glycolysis to have oxygen. It can very well function in absence of oxygen also. And now let's talk about little bit about anaerobic and slash fermentation. When we talk about anaerobic, it is absence of O2. When we talk about fermentation, it is limited supply of O2. Okay. In both the conditions, glycolysis can happen, will happen. We also saw that it happens in the cytoplasm. The pyruvate that is formed then enters into the mitochondria where the Krebs cycle will happen. Okay. So this was all about glycolysis, and I hope there are not many doubts now. Thank you.